So today we will learn chapter 5, the structure and the function of membrane proteins. So in this chapter we will learn the membrane proteins. So chapter 5 is composed of 6 subjects starting from the molecular organization of membrane. So we also cover membrane protein topology and function, and also we will learn the 7 transmembrane helices. And also we will focus on bacterial reaction center structure, and we will cover the oxygenic photosynthesis. Finally, we will learn the transmembrane metabarriers. So in this slide, I will explain the first subject, molecular organization of membrane. The basic subunit of membrane is the glycerol phospholipids. So this is the backbone of glycerol phospholipid is glycerol 3-phosphate, which is composed of the 3 carbon, and also in 2 carbon, the OH is attached into this carbon and this carbon. However, in third location, phosphate is attached in the third location of carbon. So that's why we call it as glycerol 3-phosphate. Okay, in the X position means that this is phosphate. Phosphate has the oxygen. So this location we call it as the X position. So in the X position, head group attached to the X position of glycerol 3-phosphate backbone. Okay, table 5.1 summarizes the common head group attached to the glycerol 3-phosphate backbone. The very famous one is the ethanolamine and also inositol. So in the case of the ethanolamine, okay, so ethanolamine, so the head group is the ethanolamine. So after that, this ethanolamine is attached to this X position. So we call it as this one, phosphatidyl ethanolamine. How about another example is inositol. So this is the inositol. So this head group is attached to the X position. So after they're attached to the X position, we call it as phosphatidyl inositol. Okay. So in this slide, I will explain another important structure in glycerol phospholipid. So this is the C1 position, and this is the C2 position. Okay, and C1 position, so OH group is attached to the C1 carbon, and also another OH is attached to the C2 carbon. Okay, so usually in this C1 carbon and C2 carbon, so fatty acid which has a long chain, so we call it as a long chain fatty acid, can be attached to the C1 and C2 position. Okay, so after that they will make this kind of structure and this kind of structure. Okay, so first they remove OH. After that they will add the fatty acid. Okay, they will add fatty acid. We call it as this structure as the acyl group, acyl chain. The R2 has the long chain. So in this case we call it as acyl chain. Okay, so table 5.2. okay, acyl chain has they have the double bonded oxygen atom, and also this acyl group is usually derived from the carboxyl acid. Okay, table 5.2 summarizes the common acyl chains found in the lipids of the biological membrane. Okay, the very famous one is palmitic acid. So they have the carboxyl group, and also they are composed of 16 carbon. Okay, first, second, After they have 13, and finally they have the 16. 1, 2, 3 plus the 13 carbon, total is 16. So in this acyl chain, they don't have a cis double bond. However, in the case of palmitoleic acid, they have one cis double bond. In this case, the numeric representation will be 16 to 1. It's like this. 1 means that they have one cis double bond. Okay. Another case is the linolenic acid, which they are composed of 18 carbon. 1, 2, 3, 4, and 18 carbon. And also they have the 3 cis double bond. Actually the full name of the linolenic acid is like this, and they have in 9th carbon they have the cis double bond, and 12 carbon they also have cis double bond, and 15 carbon they also have the cis double bond is like this. Okay, so the important thing in this slide is in C1 and C2 position. So usually the fatty acid, which has a long chain, we call it as the acyl chain, can be attached to usually this fatty acid, remove OH, after can be attached to C1 and C2 position. Okay, so this is the basic structure of glycerol phospholipid. Explain the characteristics of phospholipids. The first characteristics is that they are amphiphilic. So phospholipids is composed of the polar and the non-polar region. Especially the polar region is in head group region. So this is X position. In head group region, they are usually positively charged. Okay, and also they also have a non-polar region. So in this case, in non-polar region, usually the fatty acid, which has the acyl chain, is existed in the two-tail region. Okay, so in the case of the two-tail region, they are non-polar. They don't have any charges. Okay, and also if the cis double bond happens, so okay, it will induce volume increase. After that, it will affect transition temperature, fluidity, and structure. So in the case of the stearic acids, they are composed of 18 carbon, and also linolenic acid, they also are composed of the 18 carbon. Both of them is the acyl chain. They have the acyl chain. However, so until 9th carbon, their structure is exactly same. However, linolenic acid, they have in 9th carbon, they have the cis double bond, and the 12th carbon, they also have cis double bond, and 15th carbon, they finally have the cis double bonds. So in the case when we compare the volume, okay, so you can see that in the case of the linolenic acids, so volume increase happens. So this volume increase affects transition temperature and fluidity and structure. The second characteristics of phospholipid is phospholipid bilayer. So phospholipid bilayer consists of the two layers of phospholipid. So with the hydrophilic exterior, which is composed of the polar region, and also hydrophobic interior, so which is composed of non-polar region. So actually the electron density profile reveals the structure of phospholipid bilayer. So two maximum peak was observed at the head group, because the head group is composed of phosphate group and also they have several side chain, and also minimum peak in the middle of the bilayer. So because they have the tail has the acyl chain, because they have the CH3 group, so they show that minimum peak in the middle of bilayer, and also they show that minimum peak in the water layer. Okay, so once again, and also the DHH means that the basic dimension of bilayer DHH is about 4 nanometer. Okay. Okay. Once again, the second characteristics of phospholipid is phospholipid bilayer. So actually, the electron density profile reveals the structure of phospholipid bilayer, and also consists of 2 layers of phospholipid, and also hydrophilic exterior and hydrophobic interior. And also DHH dimension is about 4 nanometer. So in this slide, I will explain the determinant of phase transition temperature. We call it as Tm in phospholipids bilayer. There are 3 criteria. First one is the length of fatty acid chain, and also cis double bond, and final one is pH. Okay, so let me start the length of fatty acid chain. So okay, phospholipids composed of the head group and 2 tail group. Okay, so 2 tail group is they have the acyl chain, which is composed of CH3, CH3, CH3. Finally, they will have the carboxyl group like this. So between the each tail, so usually the van der Waals interaction happens. Okay, so if they have a long fatty acid tail, it means that van der Waals interaction is strong. So it will induce low fluidity. So if they have a short fatty acid tails, so the weak interaction happen between each tail, and also it will induce high fluidity. The short chain induced high fluidity. Finally, it will induce lower Tm, lower phase transition temperature. Okay, table 5.3 summarizes the phase transition temperature of common phospholipid bilayer. Okay, so here you will see that this is phosphatidylcholine. The first one is composed of the 22 carbon, second one is 18 carbon, 16 carbon, and 14 carbon. Okay, so when we compare the transition temperature, you will see that the transition temperature is starting from 75, 55, 71, 24. Okay, how about another cases? The phosphatidyl ethanol amine. So this phosphatidyl ethanol amine is composed of the 16 carbon, and this phosphatidyl ethanol amine is composed of 14 carbon. So you will see that transition temperature is 60, but the transition temperature in this one was 50. So it means that short chain is induced weak interactions, so that high fluidity happens. So finally they exhibit lower Tm. How about cis double bond? So if there is a cis double bond, so it will induce unsaturated fatty acid like this. Okay, so this is the unsaturated fatty acid, and this is the saturated fatty acid. Okay, in the case of when the unsaturated hydrocarbon existed, so it will the membrane, the phospholipid bilayer, the status will be the fluid. Okay, so it means that unsaturated R group it will induce high fluidity, finally it will exhibit lower TM. Okay, let me compare here. So this is the phosphatidylcholine, which is composed of the 18 carbon. So another one is also phosphatidylcholine, which is composed of 18 carbon, but one carbon they have the cis double bond. So when you compare the transition temperature, the first one is 55, but the other one is minus 22. So if there is unsaturated R group, so it will induce the high fluidity. Finally, it will exhibit lower TM. How about pH? So pH is when they were in around the pH 7, or when they were in pH 10. So high temperature, when they were in high temperature, it means that in the environment there will be loss of OH minus group. So in these cases, when they were in higher temperature, the fatty acid chain will be unstable like this. So it will induce high fluidity. So means that finally it will exhibit lower TM. Okay, so this is the curve based on when they were in the pH 5.1. The TM was around 45. When the pH is increased, when pH was 10.7, the TM was around the 40, like this. Okay, in this table 5.3, you will see that when you see the phosphatidylserine, okay, one case is in lower pH and the other case is in high pH. When they were in lower pH, around the transition temperature is 72, and when they were in the high temperature, transition temperature is 55. So it means that the high temperature exhibit lower TM. It's like this. In this slide, I will explain the second subject, membrane protein topology and function, seen through organization of the erythrocyte membranes. So what is erythrocyte? Erythrocyte is the 적혈구 in Korean, and also they have the membrane structures like this. So membrane is composed of the integral proteins which span the membrane, so like this, and also there is integral peripheral protein, and also there is a lipid linked proteins like this. Okay, because for this chapter is focused on membrane protein, so now we will focus on the integral membrane protein. Okay, let's start from the band 3 protein. So band 3 protein is this is the full length of the full length structure of the band 3 protein, starting from N terminal and C terminal. And this is the actual their structure. It's like this. Okay. They have the cytoplasmic N terminal domain, and they also have cytoplasmic C terminal domain. So let's start from the cytoplasmic N terminal domains. So in the cytoplasmic N terminal domain, they interact with the ankyrin and also protein 4.2. So they have a domain like this. So this domain interact with the ankyrin, and also this domain interact with the protein 4.2. So actually this domain interact with the ankyrin, and this domain interact with the 4.2. So the function of the cytoplasmic N domain, cytoplasmic N terminal domain, is link the membrane to the underlying cytoskeleton. Okay, this is the underlying cytoskeleton, which is composed of the alpha spectrin and beta spectrin. So actually the cytoplasmic N terminal domain is link the membrane to the underlying cytoskeleton via ankyrin and protein 4.2. Okay, so this is the cytoplasmic and terminal domain. Okay, band 3 protein also has the membrane domain. So this is membrane domain. So membrane domain spans the membrane protein times. Okay, let's see in details. They have a transmembrane domain 1, and also they have a transmembrane domain 2. Finally, there is the transmembrane domain 14 is here. Okay, so after that, so there is the C-terminal domain is like this. So in this C-terminal domain, they have the dimerization arm. So it induces, this is the band 3 protein, the one subunit. So, and one subunit, they made dimerization like this. So usually the C-terminal domain has the dimerization arm. So they induce dimerization of band 3 proteins. So main function of this dimer structure. This dimer structure is a function as anion exchanger. So first they transport HCO3 minus HCO3 ion out of the cell. After that, in exchange for chloride ion. So chloride ion can be go inside. So usually they transport HCO3 ion out of the cell, and in exchange for chloride ion is like this. Okay. The special characteristics of the integral membrane protein is it contains higher percentage of the hydrophobic amino acid, including alanine, leucine, isoleucine, valine. So table 5.5, the length, the hydrophobicity of the side chain of the amino acid. Okay, let me start alanine. Alanine, the value is 1.8 and leucine is the 3.8. How about isoleucine and valine? So they have a 4.5 and valine has a 4.2. The higher value means that their side chain is hydrophobic. If the value is minus, means that they are hydrophilic. Okay. Okay, concept check 1. So which of these residue is not expected to contact a lipid bilayer in the nonpolar region of the membrane? So valine, lysine, isoleucine, leucine, the alanine. Maybe the lysine, the value minus 3.9. This lysine is not expected to contact a lipid bilayer in the nonpolar region because they are hydrophilic. Okay, so hydropathy plot. This is hydropathy plot. Is reflect the preference of amino acid side chain for polar and non-polar environment. Okay, so if the side chain is hydrophobic, so they prefer non-polar environment. If side chain is hydrophilic, they have minor charge, so they prefer polar environment. So this is the hydropathy, the plot. So if you see the higher value, it means that there is the transmembrane domain. Okay, if there is a higher value like this, peak like this, means that there is the transmembrane domain and transmembrane domain and transmembrane domain. So actually, this is the band 3 proteins hydropathy plus. Okay, and this based on this hydropathy plot, we assume that there will be protein transmembrane domains. So in this slide, I will explain another integral membrane proteins in erythrocyte membrane. We call it as a glycoprotein A. Glycoprotein A is composed of three domains. Which one is? The first one is external N-terminal domain and membrane domain, and also cytoplasmic C-terminal domains. Okay, let me start external N-terminal domain. Okay, this is the external N-terminal domain, has many hydrophilic amino acids. So if you see here, serine, which has the OH group, threonine, which has OH group, threonine, after that, there is the threonine, which has the OH, serine OH, and also there is the lysine, which has a positively charged, and also there is the serine, threonine, asparagine, which has the NH2 group, and also threonine, aspartate has the minus charges, glutamate is minus charged. So this protein, all of them is the hydrophilic amino acid. Okay, they have the many hydrophilic amino acid. And also this external N-terminal domain is glycosylated in the serine and threonine and asparagine. Okay, this glycosylation, we call it as a post-translational modification. Serine is a polar amino acid. They have OH group. Threonine is also, they have a polar side chain which has OH group. And asparagine has the carboxyl amino group. They have this asparagine. Okay. And the O-linkage glycylation happens in the OH group in serine and threonine. And also N-linkage glycylation happens at NH group in asparagine. So this is, we call it as the O-linkage glycosylation, and this is the N-linkage glycosylation. Okay. Let me summarize it once again. The external N-terminal domain has the many hydrophilic amino acid. Okay, and also glycosylated in serine, threonine, and asparagine. Especially in the serine and threonine, O-linkage glycosylation is happened, and asparagine is N-linkage glycosylation is happens. And also glycoprotein protein, glycoprotein A is also composed of the membrane domain. Okay, this is the membrane domain. And hydropathy plot revealed that, okay, they have the one peak is like this. It means that glycoprotein A has the one transmembrane domain. And this transmembrane domain is composed of the many hydrophobic amino acids, which is the isoleucine and isoleucine, the leucine, isoleucine, valine, or alanine. It's like this. They have the that is composed of the many hydrophobic amino acid. Okay. And also, finally, there is the cytoplasmic C-terminal domain. So this is the cytoplasmic C-terminal domain, has the many hydrophilic amino acids, which is arginine, which is the positively charged, and the lysine is also positively charged. Okay, and also there is serine, and threonine and serine. It's like both of them is they have OH group. Okay, so they have many hydrophilic amino acid, but is not glycosylated. So they have for example, they have asparagine, and they also have the serine. they also have threonine, but this serine, threonine, asparagine is not glycosylated. There is no O-linked glycosylation, and there is no N-linkage glycosylation. So nearly all of scientists used erythrocyte membrane as a model for studying membrane proteins. Why? There are several advantages, because we can easily obtain blood which contains erythrocyte, and also centrifugation allows the separation of erythrocyte from other components. So this is the erythrocyte. The erythrocyte makes up about 45% of the total blood. So we can easily obtain the erythrocyte just by centrifugation. And also peripheral protein. So this is peripheral protein, and this is integral proteins. So peripheral protein can be obtained by treating hypertonic solutions. So after we treated hypertonic solutions, cell can be shrived. After we can easily extract the peripheral proteins. Okay, this is the advantage. However, they also have, the erythrocyte also have a disadvantage. For example, in the case of the integral proteins, so peripheral protein we can easily obtain by just treating hypertonic solution. But in the case of the integral protein, to isolate integral proteins, it requires many solutions, for example, detergent and also organic solvents, and also they need several mechanical fractionation methods, for example, homogenation and sonication. However, advantage is the more than the disadvantage. So people usually use erythrocyte membrane as a model for studying membrane proteins. So in this slide, I will explain the third subject, bacterial rhodopsin and the discovery of 7 transmembrane helices. So this is a bacterial rhodopsin structure. So actually, where is the bacterial rhodopsin? So bacterial rhodopsin usually observed in the membrane of the halobacterium. So it recognized light. It's like this. Okay. So now the structure is composed of 7 transmembrane helices, and also there each transmembrane helices connected by loop region. So each 7 transmembrane helices, they exhibit high content of hydrophobic amino acid, especially the alanine, leucine, isoleucine, valine, which has the aliphatic side chain. So they are hydrophobic amino acid. So if you see here, there are lots of isoleucine, leucine, alanine, and valine. Isoleucine is like this. So 7 transmembrane helices show that high content of the hydrophobic amino acid ALIV. So how about the loop region? You will see that in the loop region there are lots of charged amino acid, especially the aspartate D and glutamate E. They have a minus charged, and also lysine and arginine, they are positively charged. For example, they have the glutamate, and also they have the arginine and lysine is like this. So in loop region, they exhibit high content of the charged amino acid, especially the negatively charged and positively charged, is like this. Okay. So in this slide, I will explain the bacterial rhodopsin mechanism. So bacterial rhodopsin mechanism happens in the retinal. So this is retinal structure. So retinal comes from the beta carotene. So carrot has lots of the beta carotenes. So beta carotene is divided by 2. So it will make 2 vitamin A. So vitamin A has finally they have OH group and OH group. This OH group is changed to the aldehyde. So this is we call it as retinal. Another name is vitamin A aldehyde. So their structure is all trans isomer. So actually they bind to the lysine of the bacterial rhodopsin. So actually the specific site is 216th location. They have the lysine. For example, so this is bind to the lysine. So this O is bind to this nitrogen, this lysine. So, okay, this is they bind to the lysine of bacterial rhodopsin. So where is this lysine located? Okay, so this is the halobacteria. They have the bacterial rhodopsin here. So this is the cytoplasmic location, and this is exterior side. So they are composed of A, B, C, D, E, F, G helix. So lysine 216th location is located in the G helix. This is the lysine. So this retinal bind to the lysine. So after light absorption, so this retinal structure is changed. Okay, so all trans isomer is changed to 13 cis retinal structure. So all trans isomer after they observed the light, so this will change 13 14th carbon. So this one changes to the cis configuration. So after that they change it to cis configuration. So this 13 cis retinal can transport hydrogen proton from the cytoplasm to extracellular sites, but I will not cover in detail in this mechanism. But the important thing is they transport after they receive the light. They can transport hydrogen from cytoplasm to the extracellular site. So they can transport proton from cytoplasm to extracellular site by using the 13 cis retinal. Okay. In previous slide we learned bacterial rhodopsin. So in this slide we will cover rhodopsin. So rhodopsin is present in the eyes of the vertebrate and or invertebrate. So in the vertebrate, rhodopsin is present in the rod cell. Especially rod cell is composed of the outer segment and the inner segment. So rhodopsin is present in the out segment. And out segment is composed of the disc, each several disc. Okay, rhodopsin is present in the disc. Okay, so rhodopsin is also composed of 7 transmembrane helices. So, okay, so this is the rhodopsin structure. They also have 7 transmembrane, and also finally the retinal is attached to the G helix, A, B, C, D, E, F, G, 7th helices. Okay, so the G helix has the lysine residue, so retinal is bind to the lysine residue. Okay, and so this is interior cytoplasmic region, and this is exterior, the extracellular side. Okay, so after rhodopsin receives the signal, for example, if rhodopsin receives light, so there will be subsequent signal transduction pathways. So we will see some details. Okay, so subsequent signaling pathway is conducted by transducin. Transducin is composed of the heterotrimeric G protein. So this is heterotrimeric G protein is composed of G alpha unit and G beta unit and G gamma unit. Actually, the G alpha unit, G alpha subunit is bind to the rhodopsin. Okay, so this is the G alpha subunit. It's good. They have the alpha helix. They are composed of alpha helix, and there are some reason they are composed of beta sheet, but they don't have the transmembrane domains. They only bind to the rhodopsin. Okay. The function is that after light is there, light will become the stimulatory external signal, and the rhodopsin receive the signal. After that, rhodopsin transport H plus from cytoplasm to extracellular side. After that, there is a subsequent signal transduction pathway initiated at G alpha subunit. So initially the G alpha subunit has a GDP, but GDP and GTP exchange happen in G alpha subunit. After that, after exchange, G alpha and GTP will be made. Okay, so this is the active status. After that, the active status G alpha, GTP is separate from the G beta and G gamma dimer. Okay. After that, this active status of G alpha and GTP transport the signal to adenyl cyclase. Okay, here. So this is the adenyl cyclase. So adenyl cyclase is an enzyme anchored to the cell membrane. The main function is that it receives the signal. So adenyl cyclase, so it receives the signal from the G alpha GTP. They are in the active status. Okay, so after that, the main function of adenyl cyclase changes to ATP into CAMP. So CAMP actually plays an important role in the cell signaling. Okay, so it will induce several cellular responses, but I will not cover in details. Okay, the important thing is that after rhodopsin received external signal, okay, rhodopsin has 7 transmembrane helices, and also they have, in the G helices they have the lysine residue. In that lysine residue, usually the retinal is bind there. Okay, so actually the retinal is there, the old trans isomer. So after they observe the light, the old trans isomer is changed to the 13 cis double bond. This happens. So okay, after that, rhodopsin can transport H plus from cytoplasm to extracellular side. Simultaneously, GDP and GTP exchange happen in G alpha subunit. And G alpha and GTP, G alpha is become the GTP status. This is active status. So this active form will separated from G beta and G gamma dimer after that it transports signal to the adenyl cyclase. The main function of adenyl cyclase is receive signal from the G alpha GTP, and after that the main function is the changes the ATP to CAMP. Actually, and also CAMP plays important role in several cell signaling pathways. So it will induce cellular response. Okay, I will not cover in details, but we have to know some signal pathway important step, and also we have to know some the protein's name. Okay. The previous slide we learned that bacterial rhodopsin and the rhodopsin has 7 transmembrane helices. So in this slide we will learn another protein which has the 7 transmembrane helices. Okay, the name is the beta-2 adrenergic receptor. So actually the adrenaline binds to this receptor. And this receptor has 7 transmembrane domains. The structure is they are composed of extra N-terminal domain, and also they have a membrane domain, and they also have cytoplasmic C-terminal domains. Okay, so external N-terminal domain is they are composed of extracellular spaces. So usually the glycosylation happens in extracellular spaces, especially in the serine, threonine, and asparagine. Okay. Okay. After that, this is a transmembrane domain which have 7 transmembrane helices. These 7 transmembrane helices has higher content of hydrophobic amino acid. For example, alanine, and also leucine, isoleucine, and valine. If I call it as the lysine, if I call it as alanine and leucine, isoleucine, and valine, you have to think about all of their structure, okay? And also they have the cytoplasmic C-terminal domain. So actually this domain is bind to the G alpha subunit. G alpha subunit, okay? So okay, they also have a loop region, and this region, the loop region, this region. In this region, they have the higher content of the positively or negatively charges. For example, aspartate and glutamate is negatively charges, and lysine and arginine is positively charges. There are lots of the positively and negatively charged protein, amino acid will be here and there. Okay. The main function of beta-2 receptor is that adrenaline is bind to this receptor. After that, it transports the signal to G alpha subunit. So G alpha subunit is activated. So G alpha GDP is changed to G alpha GTP. After the active status G alpha subunit transport signal to the adenyl cyclase. After that, adenyl cyclase changes the ATP to CAMP. So CAMP did a lot of the signal transaction, plays important role in signal transaction. Signal transaction pathway. For example, they will open the calcium channel while they will open the potassium channel. I will not cover in detail in this mechanism, but important thing is that adrenaline is bind to this receptor, that it transport signal to the transducin, especially G alpha subunit, and G alpha subunit is activated. They will become G alpha GTP, After that, it will dissociate separated from G beta gamma dimer. After that, it will activate the adenyl cycloase. The adenyl cycloase changes ATP to CAMP. So CAMP plays important role internal signal, okay, signal transduction pathway. For example, they will open calcium channel or they will open potassium channel. In this slide, I will explain the post subject, the structure of bacterial reaction center. So bacterial reaction center is belong to photosynthetic complex in cyanobacterium. So cyanobacterium has the photosynthetic complex, which is composed of LH1, LH2, and LH1, and the reaction center, and the cytochrome BC1 complex. So reaction center is located here. Actually, reaction center is the inside of the light harvest LH1, light harvesting complex 1. Okay, so this is the reaction center. So before we go further, so in this slide we will briefly learn about LH1 and LH2. So LH2 and LH1 is function as antenna. So this LH2 and LH1 observe a proton, the light, okay, and release after it observes the proton, it releases 2 electrons. Okay, let me start the LH2. So this is LH2, this is LH2. So LH2 contains bacterial chlorophyll B800. So this is the B800. So this B800 observed 800 nanometer the wavelength light. So this is the structure of bacterial chlorophyll B800. So this is the LH2 structure, which is composed of the transmembrane helices, but I will not cover in details. Okay, the LH2 has the B800. B800 is that the red color is like this. So this is the bacterial chlorophyll B800. It observes 800 nanometer light. So around they observe in these wavelengths. And also they have the bacterial chlorophyll B850. So this is the B850. So this is the blue color is the B850. So this is observed around 850 nanometer light, and also the structure is nearly similar to bacterial chlorophyll 800. Okay. And also LH2 has the carotenoids. This is the orange color is carotenoids. The carotenoids usually observed around between the 400 and 500 nanometer wavelengths. Okay, and also especially LH1. LH1 is located here next to the RC, but actually the inside of LH1 there is a reaction center is located here. So LH1 contains bacterial chlorophyll B875. So this is observed around 875 nanometer wavelength light. That's right. Okay. Okay. So this is the LH1 and LH2 is the function as antenna. So main function is observe a photon. After that, it will release 2 electrons. Okay. So now we will cover, we will focus on the reaction center. So this is the reaction center. So reaction center is they have the electron transporter system. Okay. So bacterial chlorophyll is so after bacterial chlorophyll receive the protons. Okay. So okay, after that bacterial chlorophyll is become the dimer in the reaction center. Okay, so after that this dimer transport two electron to bacterial pheophytin. The purple color is bacterial pheophytin. After that, bacterial pheophytin transport two electron to quinone acceptor QA. After that, it also transport two electron to quinone acceptor QB2. So okay, after QB, quinone acceptor QB accept two electron from QA. It also receives the two proton from cytoplasm. So this is cytoplasm from cytoplasm. After that, it will synthesize QH2 quinone. It will synthesize QH2. So this is the main mechanism of a reaction center, the electron transport system from bacterial chlorophyll dimer to the quinone accept QB, and finally it received proton from cytoplasm and synthesized QH2. So this is the reaction center's main function. So we will see the detailed structure of a reaction center in the next slide. Okay. Now, okay. So in this slide, I will briefly explain the cytochrome BC1 complex. So cytochrome BC1 complex is composed of cytochrome B and cytochrome C1. Okay. So let me start the cytochrome B. So cytochrome B accepts two electrons from the QH2. Okay. So after that, cytochrome B will be activated. So the activated cytochrome B also receives the two protons from cytoplasm and synthesized QH2. Okay, how about the cytochrome C1? So cytochrome C1 is the transfer electron from the low potential QH2 and also to higher potential cytochrome C2. So one QH2 used by the cytochrome B and another QH2 can be used by cytochrome C1. So cytochrome C1 transfer two electron from low potential quinone, we call it as a QH2, to higher potential cytochrome C2. So, okay, so cytochrome, so this is the electron transfer, and this is also electron transfer. So they links electron transfer to the proton translocation. For example, 4 protons can be translocated from cytoplasm to the periplasm. Okay, so the main function of cytochrome BC1 complex is that cytochrome B is accept 2 electrons from QH2. At that, the activated cytochrome B also receives the 2H plus 2 protons from cytoplasm and synthesize QH2. And cytochrome C1 is also receives 2 protons from QH2 and also transfer the electron to the high potential cytochrome C2. So by doing this electron transfer procedure, it links proton translocation 4 proton can be translocated from cytoplasm to the periplasm. In previous slide, we learned that cytochrome C1 transport 2 electrons to cytochrome C2. And cytochrome C2 also transport 2 electrons to reaction center. So this cycle, we call it as the electron transport system, or we call it a Q cycle. Okay, so after finished electron transport system, so 4 proton is transported into from cytoplasm and the periplasm. So this is the bacteria cell. Bacteria has cytoplasm, and between the membrane and membrane there is periplasm is located here. So 4 proton is accumulated in the periplasm. So ATP synthase uses electrochemical gradients created by the difference in proton concentration between the cytoplasm and the periplasm. So finally, it will create ATP from ADP and inorganic phosphate PI. So this is I just briefly explained the ATP synthase functions. Okay. So in this slide, I will explain the bacterial reaction center structure. So this is the bacterial reaction center. RC is composed of the 4 subunits, L subunit, M subunit, H subunit, and cytochrome subunit. Okay, let me start H subunit. Okay, so H subunit is the smallest subunit. So this is the H subunit. Blue color is H subunit. So they have the transmembrane helix. So this is the membrane. Membrane is here and membrane is here. They have a transmembrane helix. It means that this area, we can expect that there are lots of the hydrophobic amino acid. Okay, and they also have a space area. So this space area bind to the L subunit and the M subunit. Okay. So in this slide, I will explain the L and M subunit. So they are two-fold symmetry structure. So when we fold it like this, they are exactly same, nearly same, like this. So they are two-fold symmetry structure. So this is the L and this is M. When we fold it like this, they are nearly same. Okay. And L subunit and M subunit, both of them is composed of 5 transmembrane helices. 1, 2, 3, 4, 5. Okay, see, for example, subunit L, they have the A, B, C, D, E, and subunit M is also composed of the A, B, C, D, E. So each is the starting from 21 amino acid, and the longest one is the 28 amino acid, like this. Okay. Usually the A three helix, for example A, B, D. So they are regular and straight helices. So A, B, D they are straight and regular helices. However, C, this is the C, and this is E, they are distorted. C is also distorted helices, and E is also distorted helices. Okay, especially helix E has the cork around here. There is a proline residue is located here. Especially helix E, a cork is caused by a proline residue around here. Okay, So helix C and E is the distorted helix. Especially helix E, there is a cork region caused by a proline residue. Okay. So in this slide, I will explain the final subunit in bacterial reaction center. So this is the cytochrome subunit, and this is cytochrome subunit, which is located in the periplasm. Okay, cytochrome subunit is composed of the 9 helices, alpha helices, 1, 2, 3, 4, 5, 6, 7, 8, 9, and they also contains 4 hem group, 1, 2, 3, 4. So main function of the cytochrome subunit is transfer electron to a bacterial chlorophyll dimer. So this is the main function, the transfer the electron to bacterial chlorophyll dimer. Okay. Okay, next. So active component is composed of the 4 bacterial chlorophyll. So this is bacterial chlorophyll dimer, and this is also bacterial chlorophyll dimer, and also 2 bacterial pH and QA, ion, and QB. So their structure, they are present. This is the L subunit and the M subunit. So between the L and the M subunit, there is a hydrophobic region is located. So this one, the active component, is located between L and M subunit. This that region is hydrophobic region. And also the active component is a two-fold symmetry structure. Okay, main function is transfer electron from bacterial chlorophyll dimer to bacterial pH, and also QA and QB. So this is the main function of the active component, which is present between the L and M subunit. Okay. So in this slide, I will explain the 5th subject, oxygenic photosynthesis. Previously, we learned photosynthesis in bacteria, especially in cyanobacteria. They are composed of LH2 and LH1 and reaction center. And also they are composed of cytochrome BC1 complex. Finally, they have cytochrome C2. And also they have ATPase. Okay, so this is the photosynthesis in the bacteria. However, oxygenic photosynthesis system is present in the algae and plant. So this is the present in the bacteria. So it means that this system is more upgraded than this system. Okay, and they are composed of the photosystem 2 and photosystem 1. So main function is they changes H2O to O2 and with ATP generation by using the ATPase. This is the same, but in this system they generate NADPH. Okay, the particular thing is that this oxygenic photosynthetic system generated O2 and also generated NADPH. But this system in bacteria, they just generated ATP. Okay, they did not make the O2 and they did not make NADPH. Okay. So in this slide, I will explain the photosystem 2 structure. Photosystem 2 is the membrane-bound complex, which is composed of D1 and D2 subunit. Okay, so the structure is nearly similar to the reaction center. Okay, how about the D1 and D2? They are composed of 5 transmembrane helices. Okay, so this is the how about L and M subunit in the reaction center. L subunit and M subunit, each one, they are composed of 5 transmembrane helices. A, B, C, D, E is like this. Okay, they also have the 5 A, B, C, D, E, 5 T1, 5 and 5 transmembrane helices. The structure is nearly similar to L and M subunit. Okay, so photosystem 2 also has the CP43 and the CP47. So this is the 6 transmembrane helices. Okay, previously reaction center in bacteria, they only have the L subunit and M subunit is transmembrane helices. However, in photosystem 2, they have additional transmembrane helices, which is the CP43 and which is CP47. Okay, so they contained 6 transmembrane helices from 1, 2, 3, 4, 5, 6. 1, 2, 3, 4, 5, 6. And also they contains chlorophyll, the antenna chlorophyll. Okay, previously we learned that in the light harvesting complex in the bacteria, so they have the antenna chlorophyll which can harvest the light. Okay, and also they function CP43 and 47 function as the light harvesting complex. So bacteria has a LH2 and LH1, but instead of LH1 and LH2, they have the CP43 and CP47. They have the 6 transmembrane helices. Okay, in this slide I will explain the photosystem 1. Photosystem 1 is composed of the PSA-A and the PSA-B and another PSA-C. And they also have several other components, but we will focus on the PSA-A and PSA-B. Okay, PSA-A and PSA-B is they have the 11 transmembrane helices. Okay, for example, PSA, the N terminal is located here and C terminal is located here. So they have the 11 transmembrane helices. PSB2 is also N terminal is located here and C terminal is located here. So they have 11 transmembrane helices. For example, PS2, they have the different protein D2 and P47 and the D1 and the CP43 is different protein. But PSA and PSB, they have the one protein, which is composed of 11 transmembrane helices. And also this one is also 11 transmembrane helices. So this is 5 C-terminal helices is similar to D1 and D2. So 5, 1, 2, 3, 4, 5 C-terminal helices similar to the D2, D1 and D2. And also 6 N-terminal helices and 6 N-terminal helices is similar to CP43 and CP47. Okay, so PS1 and PS2 is nearly similar to each other, but they have the one protein instead of two protein. Okay, so C-terminal is nearly similar to D1 and D2, and N-terminal is nearly similar to CP43 and CP47. So this region function as also the light harvesting complex. Okay. And also they have additional the cofactor but we will not cover in details about the cofactors. Okay. So the important thing in the photosystem 2, they have the D1 and D2, which function as the L subunit and the M subunit in the bacterial reaction center, and also they have the antennal chlorophyll, which function as the light harvesting complex. Okay, and also photosystem 1, their structure is nearly similar to photosystem 2, but they have the 11 transmembrane helices. Okay, C-terminal is nearly similar to D1 and D2, which function as L subunit and M subunit in the bacterial reaction center. And this N-terminal is similar to CP43 and 47. It functions as a light harvesting complex. Okay. So in this slide, I will explain the final subject, membrane proteins based on transmembrane beta barriers. Okay, let me start OMPF purine. So this is OMPF purine structure. So they are composed of 16 beta strand. Okay, so they make the barrier. This is, we call it as barrier-like structures. Okay. So they show that, because this region is the membrane, so membrane region, okay. So this is better. So this region is membrane region. So they exhibit asymmetric distribution of hydrophobic amino acid. So this is the hydrophobic amino acid, okay. So this region is the contact region with the lipid bilayer in the membrane. Okay, so especially the hydrophobic amino acid is composed of alanine, valine, isoleucine, and leucine, and phenylalanine, methionine, tryptophan, and proline. So if I call it alanine, valine, isoleucine, leucine, you have to think about all of their structure, okay? You have to memorize the structure, okay? So this is the contact region with the lipid bilayer. And they also have a KBT. So KBT is composed of the hydrophilic amino acid, so which has glutamate, which has the minus charge, and arginine, which has a positively charged, and tyrosine, which has OH group. So OH group, its tyrosine has the polar side chain, which has OH. So in this case, so KBT is composed of the hydrophilic amino acid. So main function of the OMPF purine is facilitate the entry of small polar molecule into periplasmic space. So the small polar molecule, because the KBT is composed of the hydrophilic amino acids, so only a small polar molecule can pass this purine region. So after that, the small polar molecule can be can be transferred to the periplasmic space. Is like this. This is cytoplasm, and after that, this is periplasm, and the small polar molecule can be transported into the periplasmic space. So okay, concept check. Purine are composed of the which structural feature? So purine is they have the beta barrier, the structural feature. They are composed of 16 beta strand. Okay. So OMF purine is existed as a trimeric structure. Okay, so this is monomer, and monomer. Between each monomer, hydrophobic interaction happens. Hydrophobic interaction happens between the hydrophobic region of the better. Better has the hydrophobic region. Okay, So hydrophobic interaction happens between the hydrophobic region of the metals. At that, the small polar molecule can be transported into the periplasmic region. Okay, so this is the OMF porine, which exists as trimeric structure. So in this slide, I will explain the alpha hemolysin, which has the transmembrane beta metals. Okay, so alpha hemolysin is composed of two main subunits. First subunit is transmembrane beta metals. Okay, they are composed of 16 beta strands, and also they will make the barrel-like structures like this. Okay, they also have additional in the head group. They have the heptamer, 1, 2, 3, 4, 5, 6, 7 subunits. So this is the head, and this is the tail region. Okay, the function is that they cause lysis of red blood cell. We call it as hemolysis by destroying cell membrane. Okay, so this tail group, transmembrane beta metal, can be inserted into the membrane of the red blood cell. At that, they will lysis, or they will induce the lysis of red blood cell like this. Okay, today we learned molecular organization of membrane and also membrane protein topology and function. So by using the erythrocyte membrane, and we learned bacterial rhodopsin and the rhodopsin structure. They exhibit 7 transmembrane helices. And also we focus on the bacterial reaction center structure, and also we learned oxygenic photosynthesis. Finally, we learned transmembrane beta barriers. Okay, today we will finish the class.